Hello, my name is Emily Hollister, and I'm so glad you've tuned in today to hear my presentation on viral metagenomics. I'll be discussing viral metagenomics as a tool for identifying that needle in the haystack and unlocking opportunities for discovery. I would like to thank the organizers as well for the opportunity to speak here today. Of course, we had all hoped to be in Boston for this meeting, but I'm very much looking forward to the content from all the other speakers. The organizers have put together a really exciting lineup this year. My name is Emily Hollister. I'm the Director for R&D Computational Biology at Diversigen. Diversigen is a commercial microbiome services provider. It was born out of Baylor College of Medicine. We're based in Houston, Texas, located in the heart of the Texas Medical Center. We are a comprehensive microbiome services organization offering services from project consulting through various forms of sequencing and qPCR through bioinformatics, interpretation, and beyond. We've been around since late 2014 and have a highly talented and experienced staff of scientists, including more than 12 PhDs. We also maintain a very close relationship with our roots at Baylor College of Medicine, including the Center for Metagenomics and Microbiome Research at Baylor. As many of you may know, Diversigen is now a part of the OraSure Technologies family of companies. We were acquired in late 2019 by OraSure and now work in close relationship and close cooperation with our sister companies, DNA Genotek and Core Biome, making us a one-stop shop for all of your microbiome needs. As scientists, we certainly understand the discovery cycle and the importance of end-to-end planning and for successful study execution. Now more than ever, metagenomics has a key role to play in the development of microbiome-based diagnostics and therapeutics with the goal of translating discoveries to the clinic and perhaps to daily life. This is also a familiar paradigm for many of you, the idea of the metagenomics discovery cycle, where one might start, for example, by comparing the microbiota of healthy and diseased individuals, identify key organisms or genes associated with health and disease, and through that process of discovery, develop potential application solutions or diagnostics that could lead to new bioactive molecules, new treatments, or potential diagnostic approaches. However, this paradigm also, or often rather, ignores the fact that to date, most microbiome studies have focused on bacteria. Despite this, one could just as easily substitute fungi or viruses for nearly any step in this process. And we are certainly seeing this as an emerging trend in the literature, where it may not necessarily be that it's a single bacterium or community of bacterium, but it could be viruses or a virus-bacterial association that ultimately leads to the phenotype or outcome of interest. Now, more than ever, our collective attention is focused on viruses, or at least one particular virus. But viral metagenomics is a field that certainly deserves our attention. If any of you attended this meeting last year, you heard me give a talk on viral metagenomics and a similar brief introduction. Viral metagenomics is a fascinating field. We know that viral taxa are found throughout the body. At any site where there are bacteria, we find viruses as well, especially phage. They outnumber our gut bacteria by about a factor of 10 to 1. And we know that they vary in the context of health and disease, even in the absence of known pathogens. There is a growing body of literature demonstrating the ability of the virome to influence the immune system and immune responses, and virus-mediated approaches, especially phage therapies, are showing promise in treating antimicrobial resistant infections and other diseases and other illnesses where traditional first-line therapeutics just aren't providing solutions. Additionally, we are seeing or finding that viruses and the virome may provide answers to microbially mediated phenomena where bacterial studies have just come up short. Also, a year ago, few of us would have predicted that we would be in our current situation. As I finished my presentation last year, I left off with this slide, specifically as a call out to encourage the audience to watch this space. Really, it was meant to be a call out to the fact that there were a number of very interesting virome studies in the publication pipeline. As I mentioned, and as I'm sure you're all thinking, very few of us would have expected the outbreak and spread of a pandemic virus. And despite the crash courses that we've all received in virology and epidemiology of late, my goal today is to share some of the latest news in viral metagenomics and continue to persuade you that this is a space worth watching, considering, and potentially participating in. Although bacterial studies have accounted for the lion's share of metagenomic studies in the last nearly 20 years, virome publications are steadily growing in number. You wouldn't necessarily know it by the slope of this green line in the graph relative to the crush of bacterial focused microbiome studies that have been published since about 2000. But if we zoom in on that line, what we find is that it is a growing trend. And there are a number of reasons why viral metagenomics studies have lagged behind bacterial metagenomics studies. As I mentioned, there are a small but growing number of publications, but part of the reason for this slower growth viromes, viral metagenomics has to do with the number of challenges associated with studying the virome. Virology has been around for a long time, but in terms of global studies of the virome or broad studies, untargeted ones, it really is more difficult. Viruses can be either DNA based or RNA based. There are no universal amplicon targets, such as 16S, that made bacterial metagenomics an easier jumping off point. We know that there are substantial pathogen biases in the databases and very incomplete databases. Additionally, when we do broad sequencing of viral communities, even in the presence of enrichment methods, we still see a lot of signal coming from both host and bacteria. So it still remains very much a needle in the haystack paradigm when looking for new viruses. Although I hope to convince you today that it's still a worthwhile and worthy thing to do. Zooming in on this plot, you'll see that viral metagenomics is growing and has a similar curve to that which we've seen with bacterial metagenomics. And certainly in light of all of the recent developments around the new coronavirus, I fully expect to see more virome studies taking place. Um, Part of the reason for this is, of course, increased awareness, but of course, improvements in nucleic acid isolation techniques have helped with this. The rapid decrease in next-gen sequencing costs, and the increasing recognition of the influence of viruses both on the host and on members of the microbiome. Very interestingly, some of the major discovery applications for RNA-seq viruses are coming in RNA-seq data sets, specifically those that have sought to look more at host, but have also captured viruses as a bycatch or as a byproduct of that effort. So as people continue to recognize this, continue to identify the need and the importance around viral surveillance, I fully expect that we'll continue to see this field growing and emerging. And that's certainly supported by other publications. It's true of both DNA viruses and RNA viruses. And this was a paper published about 2 years ago, highlighting the fact that uh, RNA viral metagenomics is still growing rapidly and is certainly not enough to cover the variety of viruses that are out there, specifically highlighting the rise of RNA metagenomic pipeline or publications, virome publications, and the rapid growth of recognized families, genera, and species in the viral taxonomy. And this really contributes to a discovery utility cycle whereby the increased utility of metagenomics leads to increased amounts of information, better search tools, more information, better databases, increased discovery, and so becomes a self-feeding cycle. So how is the field utilizing viral metagenomics? In my talk today, I'd like to highlight some of the latest work from the field of viral metagenomics and demonstrate the ways in which this data and these approaches are being utilized. Throughout the next several slides, you'll notice these icons placed as an indicator of the ways in which data are being used. For example, just as in the early days of bacterial metagenomics, many viral metagenomics studies have a natural history or cataloging type focus to them. That is, discovering what is present, where, and when. Others jumped ahead of this step and are seeking to identify relationships between viruses, and especially phage, and health and disease. Others are looking to viruses where other diagnostic approaches have come up short, specifically in the context of clinical metagenomics. And still others are using viral metagenomics as a pathogen detection and surveillance tool. As we've all seen, this can have important health implications, but it's also can be a key mechanism for getting one's discovery work funded. First, I'd like to talk about some of the natural history type applications. Recently, there was a paper published in the water quality literature proposing the potential to use viral metagenomics or viruses as a means for water quality assessment and water quality tracking. For years, fecal indicator bacteria have been used for this purpose, but we know that fecal indicator bacteria are not good indicators for viral pathogens. Likewise, we know that it can be difficult to necessarily pinpoint sources of fecal bacteria, whereas viruses can be more host specific. And so if one is specifically looking to distinguish human versus farm animal or wildlife contributions to water quality or decreasing water quality, viruses can provide better resolution than bacteria sometimes. Similarly, knowing what is present under normal or baseline conditions can give us a reference for the interpretation of new information. Viral applications for clinical metagenomics may be on the horizon as the field improves its ability to provide clinically relevant turnaround times. And this is an slightly older study that highlights what's present in the blood DNA virome in a large number of individuals. So it's not uncommon in humans, in human blood specimens, to find herpes viruses, anelloviruses, and other viruses, including things like HIV or influenza, that may reflect variation with respect to age, sex, ancestry, biogeography, recent immunization, and unfortunately, in some cases, reagent contamination. Bringing this guide up again, and I'll transition to highlight some recent studies that have identified links between the virome, health, and disease. We've learned in recent years that the gut virome can reflect birth mode longer than bacterial signatures. In this study published out of the microbiome group at Cork, what they found was that even a year after birth, the signature of vaginal versus cesarean birth was present in the virome, whereas infants could be distinguished fairly readily, whereas it no longer persisted in the 16S-based bacterial signature. And specifically, although no virus was unique to birth mode, 32 of them were differentially abundant, including bifidobacterium phages. Although the implications for this are not known, it's certainly an intriguing finding. Recent evidence also suggests that the fecal virome shifts, as might be expected, with a change in diet composition. What's unexpected about this is that the shift is expected to be more virulent toward the host, as well as host bacteria. Both phage and eukaryotic viruses were found to shift in concert with exposure to a high-fat diet, and prolonged exposure at that, suggesting that the impact of dietary changes runs deeper than may have been previously expected. Recent work has also found that extended exposure to enterovirus V in early life may be a risk factor for the development of islet autoimmunity in children at risk for the development of type 1 diabetes. This was a study published late last year from the TEDdy cohort, which stands for environmental determinants of type 1 diabetes in the young, specifically a large cohort of children at risk for developing type 1 diabetes was followed through their first several years of life, looking at conversion to islet autoimmunity and ultimately type 1 diabetes. And what this group found was that extended exposure to enterovirus B was a risk factor, an increased risk for development of this autoimmunity. Although viral exposure is a trigger for type 1 diabetes has been previously hypothesized, the evidence for it has been mixed. But this work suggests that it may be a specific enterovirus rather than viral exposure in general. Next, I'd like to highlight some studies and clinical applications and their association with health and disease. Again, bringing up this DNA virome, blood DNA virome study, um, where it certainly is important to be able to have reference ranges for the interpretation of clinical outcomes. More interest or more current to that is a study based on 2 years of applications of viral metagenomics in a tertiary diagnostics unit in Europe. What was really interesting about this study was that this group utilized viral metagenomics to confirm some viral infections, and they were able to identify additional viral pathogens that weren't picked up by standard diagnostic assays across a wide variety of samples and referring departments and applications, suggesting and certainly demonstrating that there is utilization or rather utility and application for viral metagenomics in the clinic, especially in the case of illnesses and infection that have no other explanation. And beyond that, this is an older study that I'm highlighting here, but certainly demonstrates the importance of screening both fecal microbiome transplantation product as well as blood products in the light of new pathogens, particularly the novel coronavirus, but also other potential pathogenic and commensal viruses are present in stool, and their transfer through FMT highlights a case for extended screening of donors in this process. Finally, I'd like to move on to veterinary medicine and surveillance activities and summarize this by saying that viral metagenomics has been key to these fields for many years. There's a large body of literature in both these fields, both veterinary medicine and surveillance. And projects like the Global Virome Project, the Predict Project, and NextStrain serve as rallying points as well as repositories for novel strains, serving as a resource for both discovery and surveillance. So this field has been growing, but as I mentioned earlier, much of the reason that the field of viral metagenomics lags behind bacterial metagenomics has been due to the availability of tools, techniques, and resources, i.e. challenges that have limited the rapid growth, or at least the same rapid growth that we have seen for bacterial metagenomics. Although we don't have perfect solutions in place today, new developments continue to help push the science forward. Specifically, mock communities are one tool that mock communities and other benchmarks are a tool that are helping in this space. In 2019, the ATCC released viral mock community products in both nucleic acid and particle format. Prior to this, one had to homebrew a viral mock community in order to test tools and techniques like extraction techniques, like sequencing techniques, or classification tools to understand the pros, the cons, the weaknesses of the new approaches being developed. Another example of this is classification tools. And specifically what we're learning, and the field has appreciated for a while, is that both nucleotide and protein search enhance classification. Viral mock communities are essential for benchmarking these tools, but we know that given the potential for viruses to mutate rapidly, translated searches are key to increased recovery of information and increased accuracy. And this is particularly highlighted here in the release of the Kraken 2X package, where translated search increases viral accuracy substantially relative to a nucleotide-based search alone. And this can be seen here as well from the Virome publication, where a tool that leveraged both nucleotide and protein search resulted in both higher precision recall and overall F-score than some of the other classifiers that are currently available in the field. I'd like to highlight the fact that nucleic acid extraction and sequencing approach matter too. I'll be showing a pair of case studies here to demonstrate that fact. So it's well known that nucleic acid extraction efficiencies vary from organism to organism and kit to kit in the context of bacterial metagenomics, and the same is true for viruses. Similarly, of course, to evaluate or to in order to be able to study RNA viruses, your sequencing approach matters too. So what I'm presenting here is a sample that was sequenced both as a WGS, that is DNA extraction only through a typical shotgun metagenomics sequencing application, or through a viral metagenomics application at Diversigen, where we leverage DNA sequencing with a cDNA step so that we capture both DNA viruses and RNA viruses simultaneously. And what you'll notice here is relative detection of various viruses and phage. And specifically, I've highlighted RNA viruses here on top. And you'll notice that we capture RNA viruses where we have that cDNA step, but not necessarily in the WGS libraries. You may look at this list of viruses and say, hey, gee, that's really interesting. This person eats fruits and vegetables or does a pretty good job at consuming things like pepper and tomatoes. What you may also notice here is the detection of tobacco mosaic virus, which indicates this person may be a smoker. So I call this the party tricks of viral metagenomics, but getting more serious about it in application, we can also look at this in the context of pathogen detection. This is a case study similarly sequenced with both DNA-only shotgun metagenomic library and a combined DNA-RNA library through our viral metagenomics pipeline. And in this case, we detect norovirus, which may explain why this person could have been ill. Likewise, sequencing depth matters, as does viral carriage and titer. This data here was work that we did confirming that our viral metagenomic classifier could detect and recover the novel coronavirus. And we found, not surprisingly, that our ability to recover the virus was a function of both sequencing depth and assumed viral load. Each of these was a confirmed positive case. This is data from the public sphere. And as I mentioned, it can be a lot like looking for a needle in the haystack. Interestingly, what we see here is that if we go from a depth of 1 GB here at the top to 18.4 at the bottom, we see a wide range in recovery of the novel SARS coronavirus 2 genome, ranging from 83% coverage to 100% coverage of the genome, and depth of coverage ranging from 3.5x to well over 100x. You'll notice that the y-axis on each of these graphs varies dramatically from roughly 20x coverage at the top of the first graph to 350x coverage at the bottom. Despite enrichment techniques that are available, viruses often still make up a small fraction, but an important minority of the data recovered in these libraries. I'd also highlight here that nearly 60%, in some cases nearly 70% of the reads recovered in the sequencing library, according to analysis provided by NCBI SRA, was human. So depth of coverage rather certainly does matter. For those who are interested, these were RNA-seq libraries of bronchial alveolar lavage taken from known coronavirus positive cases collected in China. So with that, I'll leave you with some summary slides and some summary points of where do we go from here? What's on the horizon for Virome research? We know that this field is expanding rapidly and will continue to grow as awareness of the virome shapes the way we think about health, disease, clinical applications, and global health. Certainly, continued improvements to collection, stabilization, and extraction approaches will help with with these goals. New reagents and approaches are always on the horizon, and certainly with all of the work being done around coronavirus, I expect that we'll see novel reagents, extractions, and collection approaches emerging, partly out of need, partly out of creativity, and potentially more robust for field application. We certainly expect to see expansion of viral databases and surveillance activities. Projects that may not have been funded previously because there was no perceived need may now have a renewed appreciation from both the public and funding bodies. Certainly, we expect to see additional discovery of novel multi-kingdom interactions. I didn't highlight many of them on my slides today, but certainly the interplay between bacteria, viruses, and host in the context of health and disease is providing new light on how our bodies interact with the world around them and how we navigate states of health and disease. We certainly expect to see continued development and refinement of tools, techniques, and resources, and that layering of virome information onto metagenomics studies will provide new biological insight. Many people are now appreciating the potential for viruses to act as confounding factors in clinical trials. And I fully suspect that many will consider adding this as an endpoint, a secondary endpoint perhaps, as they look at the way in which their subjects interact or come to trials with virus, as well as the way that their treatments may impact or interact with viruses that are present. And as before, I would encourage everyone to continue to watch this space. It's developing rapidly and certainly shaping our understanding of biology and medicine. So with that, I would like to thank each of you for attention, the organizers for the opportunity to speak today, and would welcome any questions. Thank you so much.